Sterile insect technique. The sterile insect technique, SIT, is a form of pest control that uses ionizing radiation, gamma ray or X-ray, to sterilize male flies that are mass-produced in special rearing facilities. The sterile males are released systematically from the ground or by air in CC-infested areas, where they mate with wild females, which do not produce offspring. As a result, this technique can eventually eradicate populations of wild flies. SIT is among the most environmentally friendly control tactics available, and is usually applied as the final component of an integrated campaign. It has been used to subdue the populations of many other fly species including the medflea, Cerathitis capitata. The sustainable removal of the tsetsefly is in many cases the most cost-effective way of dealing with the TNT problem, resulting in major economic benefits for subsistence farmers in rural areas. Insecticide-based methods are normally very ineffective in removing the last remnants of tsetse populations, while, on the contrary, sterile males are very effective in finding and mating the last remaining females. Therefore, the integration of the SIT as the last component of an area-wide integrated approach is essential in many situations to achieve complete eradication of the different tsetse populations, particularly in areas of more dense vegetation. A project that was implemented from 1994 to 1997 on the island of Unguja, Zanzibar, United Republic of Tanzania, demonstrated that, after suppression of the tsetse population with insecticides, SIT completely removed the Glossina austeni newstead population from the island. 48-49 This was carried out without any understanding of the population genetics of GA, but future SIT efforts can benefit from such preparation. Population genetics would help to select the Glossina population to be deployed for similarity to the target population. 50 The eradication of the tsetsefly from Unguja Island in 1997 was followed by the disappearance of the AAT, which enabled farmers to integrate livestock keeping with cropping in areas where this had been impossible before. The increased livestock and crop productivity and the possibility of using animals for transport and traction significantly contributed to an increase in the quality of people's lives. 51-52 Surveys in 1999, 2002, 2014, and 2015 have confirmed this success. Continued absence of tsetse and nagana on the island. 53 In the Niayes region of Senegal, a coastal area close to Dakar, livestock keeping was difficult due to the presence of a population of Glossina palpalis gambiensis. Feasibility studies indicated that the fly population was confined to very fragmented habitats, and a population genetics study indicated that the population was genetically isolated from the main tsetse belt in the southeastern part of Senegal. After completion of the feasibility studies 2006-2010, an area-wide integrated eradication campaign that included an SIT component was started in 2011, and by 2015, the Niayes region had become almost tsetsefly-free. This has allowed a change of cattle breeds from lower-producing trypanotolerant breeds to higher-producing foreign breeds. 54-55 The entire target area, Block 1, 2 and 3, has a total surface of 1,000 square kilometers, 390 square miles, and the first block, northern part, can be considered free of tsetse, as intensive monitoring has failed to detect since 2012 a single wild tsetsefly. The prevalence of AAT has decreased from 40 to 50% before the project started to less than 10% to date in Blocks 1 and 2. Although insecticides are being used for fly suppression, they are applied for short periods on traps, nets and livestock, and are not spread into the environment. After the suppression activities are completed, no more insecticide is applied in the area. The removal of trypanosomosis will eliminate the need for constant prophylactic treatments of the cattle with trypanocidal drugs, therefore reducing residues of these drugs in the dung, meat and milk. The main beneficiaries of the project are the many smallholder farmers, the larger commercial farms and the consumers of meat and milk. According to a socioeconomic survey and benefit cost analysis, 56 after eradication of the tsetse farmers will be able to replace their local breeds with improved breeds and increase their annual income by 2.8 million euros. In addition, it is expected that the number of cattle will be reduced by 45%, which will result in reduced environmental impacts. Societal impact. In the literature of environmental determinism, the tsetse has been linked to difficulties during early state formation for areas where the fly is prevalent. A 2012 study used population growth models, physiological data, and ethnographic data to examine pre-colonial agricultural practices and isolate the effects of the fly. A tsetse suitability index was developed from insect population growth, climate and geospatial data to simulate the fly's population steady state. An increase in the tsetse suitability index was associated with a statistically significant weakening of the agriculture, levels of urbanization, institutions and subsistence strategies. Results suggest that the tsetse decimated livestock populations, forcing early states to rely on slave labor to clear land for farming, and preventing farmers from taking advantage of natural animal fertilizers to increase crop production. These long-term effects may have kept population density low and discouraged cooperation between small-scale communities, thus preventing stronger nations from forming. The authors also suggest that under a lower burden of tsetse, Africa would have developed differently. Agriculture, measured by the usage of large domesticated animals, intensive agriculture, plow use and female participation rate in agriculture, as well as institutions measured by the appearance of indigenous slavery and levels of centralization, would have been more like those found in Eurasia. Qualitative support for this claim comes from archaeological findings, e.g., Great Zimbabwe is located in the African highlands where the fly does not occur, and represented the largest and technically most advanced pre-colonial structure in southern sub-Sahara Africa. 57. Other authors are more skeptical that the tsetsefly had such an immense influence on African development. One conventional argument is that the tsetsefly made it difficult to use draft animals. Hence, wheeled forms of transportations were not used as well. While this is certainly true for areas with high densities of the fly, similar cases outside CC-suitable areas exist. While the fly definitely had a relevant influence on the adoption of new technologies in Africa, it has been contended that it does not represent the single root cause. 58. History. According to an article in the New Scientist, the depopulated and apparently primevally wild Africa seen in wildlife documentary films was formed in the 19th century by disease, a combination of rinderpest and the tsetse-fly. Rinderpest is believed to have originated in Asia, later spreading through the transport of cattle. 59. In 1887, the rinderpest virus was accidentally imported in livestock brought by an Italian expeditionary force to Eritrea. It spread rapidly, reaching Ethiopia by 1888, the Atlantic coast by 1892 and South Africa by 1897. Rinderpest, a cattle plague from Central Asia, killed over 90% of the cattle of the pastoral peoples such as the Maasai of East Africa. In South Africa, with no native immunity, most of the population, some 5.5 million domestic cattle, died. Pastoralists and farmers were left with no animals, their source of income, and farmers were deprived of their working animals for plowing and irrigation. The pandemic coincided with a period of drought, causing widespread famine. The starving human populations died of smallpox, cholera, and typhoid, as well as African sleeping sickness and other endemic diseases. It is estimated that 2/3 of the Maasai died in 1891. 60. The land was left emptied of its cattle and its people, enabling the colonial powers Germany and Britain to take over Tanzania and Kenya with little effort. With greatly reduced grazing, grassland turned rapidly to bush. The closely cropped grass sward was replaced in a few years by woody grassland and thornbush, ideal habitat for tsetse-flies. Wild mammal populations increased rapidly, accompanied by the tsetse-fly. Highland regions of East Africa which had been free of tsetse-fly were colonized by the pest, accompanied by sleeping sickness, until then unknown in the area. Millions of people died of the disease in the early 20th century. 60. Serengeti National Park, Tanzania. The areas occupied by the tsetse-fly were largely barred to animal husbandry. Sleeping sickness was dubbed the best game warden in Africa by conservationists, who assumed that the land, empty of people and full of game animals, had always been like that. Julian Huxley of the World Wildlife Fund called the plains of East Africa a surviving sector of the rich natural world as it was before the rise of modern man. 60. They created numerous large reserves for hunting safaris. In 1909, the newly retired President Theodore Roosevelt went on a safari that brought over 10,000 animal carcasses to America. Later, much of the land was turned over to nature reserves and national parks such as the Serengeti, Masai Mara, Kruger and Okavango Delta. The result, across eastern and southern Africa, is a modern landscape of man-made ecosystems, farmland and pastoral land largely free of bush and tsetse-fly, and bush controlled by the tsetse-fly. 60. Although the colonial powers saw the disease as a threat to their interests, and acted accordingly to bring transmission almost to a halt in the 1960s, 610174 this improved situation led to a laxity of surveillance and management by the newly independent governments covering the same areas, and a resurgence that became a crisis again in the 1990s. 610174, 610175. Current situation. Tsetse-flies are regarded as a major cause of rural poverty in sub-Saharan Africa, 10 because they prevent mixed farming. The land infested with tsetse-flies is often cultivated by people using hoes rather than more efficient draft animals because Ngana, the disease transmitted by tsetse, weakens and often kills these animals. Cattle that do survive produce little milk, pregnant cows often abort their calves, and manure is not available to fertilize the worn-out soils. Tsetse-fly from Burkina Faso. The disease Ngana or African animal trypanosomiasis, AAT, causes gradual health decline in infected livestock, reduces milk and meat production, and increases abortion rates. Animals eventually succumb to the disease. Annual cattle deaths caused by trypanosomiasis are estimated at 3 million, reducing annual cattle production value by $600 million United States dollars minus $1 United States dollar and 20 cents B. 10. This has an enormous impact on the livelihood of farmers who live in tsetse-infested areas, as infected animals cannot be used to plow the land, and keeping cattle is only feasible when the animals are kept under constant prophylactic treatment with trypanocidal drugs, often with associated problems of drug resistance, counterfeited drugs, and suboptimal dosage. The overall annual direct loss potential in livestock and crop production was estimated at 4.5 billion United States dollars, 62, 4.75 billion United States dollars. 10. The tsetse-fly lives in nearly 10 million square kilometers, 4 million square miles, in sub-Saharan Africa. 10, mostly wet tropical forest, and many parts of this large area is fertile land that is left uncultivated, a so-called green desert not used by humans and cattle. Most of the 38 countries, 10, infested with tsetse are poor, debt-ridden and underdeveloped. Of the 38 tsetse-infested countries, 32 are low-income, food deficit countries, 29 are least developed countries, and 30 or 34 are among the 40 most heavily indebted poor countries. Eradicating the tsetse and trypanosomiasis (TNT) problem would allow rural Africans to use these areas for animal husbandry or the cultivation of crops and hence increase food production. Only 45 million cattle, of 172 million present in sub-Saharan Africa, are kept in tsetse-infested areas but are often forced into fragile ecosystems like highlands or the semi-arid Sahel zone, which increases overgrazing and overuse of land for food production. In addition to this direct impact, the presence of CC and trypanosomiasis discourages the use of more productive exotic and crossbred cattle, depresses the growth and affects the distribution of livestock populations, reduces the potential opportunities for livestock and crop production, mixed farming through less draft power to cultivate land and less manure to fertilize, in an environment-friendly way, soils for better crop production, and affects human settlements. People tend to avoid areas with CC flies. CC flies transmit a similar disease to humans, called African trypanosomiasis, human African trypanosomiasis, HAT, or sleeping sickness. An estimated 63 million people in 20 countries are at different levels of risk, and only 3 to 4 million people are covered by active surveillance. 10. The Daily Index, Disability Adjusted Life Years, an indicator to quantify the burden of disease, includes the impact of both the duration of life lost due to premature death and the duration of life lived with a disability. The annual burden of sleeping sickness is estimated at 2 million dailies. Since the disease tends to affect economically active adults, the total cost to a family with a patient is about 25% of a year's income. 64. History of study. In East Africa, C.F.M. Swinnerton played a large role in the first half of the 20th century. Swinnerton did much of the earliest CC ecology research. 65. Forthus E. E. Austin named a patronymic taxon for him, G. Swinnertoni, in 1922. 22. Resistance to trypanosomes. CC flies have an arsenal of immune defenses to resist each stage of the trypanosome infectious cycle, and thus are relatively refractory to trypanosome infection. 66. Among the host flies' defenses is the production of hydrogen peroxide. 67. A reactive oxygen species that damages DNA. These defenses limit the population of infected flies. See also. David Bruce, microbiologist. Drosophilidae. The Drosophilidae are a diverse, cosmopolitan family of flies, which includes species called fruit flies, although they are more accurately referred to as vinegar or pumice flies. 1. Another distantly related family of flies, Tephrididae, are true fruit flies because they are frugivorous, and include apple maggot flies and many pests. The best-known species of the Drosophilidae is Drosophila melanogaster, within the genus Drosophila, also called the fruit fly. Drosophila melanogaster is used extensively for studies concerning genetics, development, physiology, ecology and behavior. Many fundamental biological mechanisms were discovered first in D. melanogaster. 2. The fruit fly is mostly composed of postmeiotic cells, has a very short lifespan, and shows gradual aging. As in other species, temperature influences the life history of the animal. Several genes have been identified that can be manipulated to extend the lifespan of these insects. Additionally, Drosophila subobscura, also within the genus Drosophila, has been reputed as a model organism for evolutionary biological studies. 3. Along with D. sacchalia for the evolution of host specialization on the toxic noni fruit. 4. And Scaptomesa flava for the evolution of herbivora and specialist on toxic mustard leaves. 5. Economic significance. Generally, Drosophilids are considered to be nuisance flies rather than pests, since most species breed in rotting material. Zaprionis indianus is unusual among Drosophilidae species in being a serious, primary pest of at least one commercial fruit, figs in Brazil. 6. Another species, Drosophila suzuki, infests thin-skinned fruits such as raspberries and cherries and can be a serious agricultural pest. 7. The leaf mining Scaptomesa flava, which is nested in the genus Drosophila phylogenetically, is an obligate leaf miner of mustard plants, including the model plant Arabidopsis thaliana. 5. And is a major pest of salad brassicas in New Zealand and an emerging pest. 7. The leaf mining Scaptomesa flava, which is nested in the genus Drosophila phylogenetically, is an obligate leaf miner of mustard plants, including the model plant Arabidopsis thaliana. 5. And is a major pest of salad brassicas in New Zealand and an emerging pest of canola in the UK. 8. Drosophila repleta larvae inhabit drains and spread bacteria. Fruit flies in general are considered as a common vector in propagating acetic acid bacteria. 9. In nature. This often ruins the alcohol fermentation process and can ruin beer or wine by turning it into vinegar. There are sinking traps available on the market for this nuisance, but one quick way to strongly limit the extent of it is to vacuum clean the flies both at rest and in their slow flight. The diagnostic characteristic. This often ruins the alcohol fermentation process and can ruin beer or wine by turning it into vinegar. There are sinking traps available on the market for this nuisance, but one quick way to strongly limit the extent of it is to vacuum clean the flies both at rest and in their slow flight. The diagnostic characteristics for Drosophilidae include the presence of an incomplete subcostal vein, 2 breaks in the costal vein, a small anal cell in the wing, convergent postocellular bristles, and usually 3 frontal bristles on each side of the head, 1 directed forward and the other 2 directed rearward. More extensive identification characteristics can be found in Drosophila, A Guide to Species Identification and Use, by Teresa Marco and Patrick O'Grady, Academic Press, 2005, ISBN 0-12473052. There is evidence to support that pathogens living within certain flies are beneficial to the behavior and survival of the host. One such example of this is in the fly Scaptomesa flava, which carries the pathogen Pseudomonas syringae in exchange for the pathogen damaging the anti-herbivore defenses of plants in the family Brassicaceae, which are the main food source for the fly. There is evidence to support that pathogens living within certain flies are beneficial to the behavior and survival of the host. One such example of this is in the fly Scaptomesa flava, which carries the pathogen Pseudomonas syringae in exchange for the pathogen damaging the anti-herbivore defenses of plants in the family Brassicaceae, which are the main food source for the fly. The family contains more than 4,000 species classified. Under 75 genera. Recently, a comprehensive phylogenetic classification of the genera based on both molecular and morphological characters has been published. Lordiphosa andalusica. Scientific classification domain: Eukaryota kingdom, Animalia phylum, Arthropoda class, Insecta order, Diptera superfamily, Ephydroidea. Family: Drosophilidae. Rondani, 1856. Psychodidae, also called drain flies, sink flies, filter flies, tube sewer flies, or sewer gnats, is a family of true flies. Some genera have short, hairy bodies and wings, giving them a furry, moth-like appearance, hence one of their common names, moth flies. Two members of the subfamily Phlebotominae, which are hematophagus, feed on blood, may be called sandflies in some countries, although this term is also used for other unrelated flies. There are more than 2,600 described species worldwide, most of them native to the humid tropics. This makes them one of the most diverse families of their order. 3. Drain flies sometimes inhabit plumbing drains and sewage systems, where they are harmless, but may be a persistent annoyance. 4. Life cycle. Live drain fly larva. Datsia bispina holotype in Burmese amber. The larvae of the subfamilies Psychodidae, Psychoricinae and Horielinae live in aquatic to semi-terrestrial or sludge-based habitats, including bathroom sinks. 5. Where they feed on bacteria and can become problematic. The larvae of the most commonly encountered species are nearly transparent with a non-retractable black head and can sometimes be. The larvae of the subfamilies Psychodidae, Psychoricinae and Horielinae live in aquatic to semi-terrestrial or sludge-based habitats, including bathroom sinks. 5. Where they feed on bacteria and can become problematic. The larvae of the most commonly encountered species are nearly transparent with a non-retractable black head and can sometimes be seen moving along the moist edges of crevices in shower stalls or bathtubs or submerged in toilet water. The larval form of the fly is usually between 4 and 5 mm, 0.16 and 0.20 in long, and is shaped like a long, thin, somewhat flattened cylinder. The body lacks prolegs, but the body segments are divided into a series of rings called annuli, singular as annulus. Some of these rings will have characteristic plates on the dorsal side. The larval thorax is not significantly larger than the abdomen, giving the larva a more worm-like appearance than those of most aquatic insects. In some species, the larvae could adhere themselves to surfaces of their environment using attachment discs on their ventral side. Like mosquito larvae, they cannot absorb oxygen through water, and instead breathe via a small dark tube, a spiracle on their posterior end. They must regularly reach the surface to obtain oxygen. The larval stage lasts for between 9 and 15 days, depending on species, temperature, and environment. There are 4 instar stages. 4, 6, 7, in small numbers. The larvae are sometimes considered beneficial, as their strong jaws can cut through the hair and sludge waste in drains which might otherwise form clogs. However, unless this sludge layer is removed entirely, the adult flies will continue to find it and lay more eggs. While the biting midges also have larvae that have no prolegs and which also have attachment discs, the larvae of the net-winged midges can be distinguished from those of the moth fly by the multiple deep lateral constrictions of the latter. 6. The pupal stage lasts between 20 and 40 hours. During this stage, the insect does not feed, but stays submerged near the water surface, still breathing through a spiracle, and soon metamorphoses into an adult fly, which bursts through a seam in the pupal casing and emerges onto the water's surface. The adults are half as long as the larva, but are much broader in appearance, with a pair of hairy wings held pitched roof-like over the body. The wings have the most elementary venation of any of Psychodidae. Temporal range. Male Clogmia albipunctata, a moth-like dense coat of small hairs gives rise to the term moth fly. Scientific classification domain: Eukaryota kingdom, Animalia phylum, Arthropoda class, Insecta order, Diptera suborder, Nematocera infraorder, Psychodomorpha superfamily, Psychodoidea family, Psychodidae. Newman, 1834, 1. The Diptera, having little more than a series of parallel veins without crossveins. The adults are typically nocturnal, though they orient themselves around lights and may appear to be attracted to light and odors. They are erratic flyers, and are often seen walking or running rapidly as well as taking flight. 8. They are most active at night, but may also be seen during daylight, or near windows, lights, or illuminated display panels. 9. The adults live for about 20 days, during which they will breed only once, often within hours of emerging from their pupal casings. Females will lay their eggs between 30 and 100, just above the water line inside moist drains. Within 48 hours these eggs hatch into drain worms, the larval form. 10. Psychodidae SEM top view. Psychodidae SEM rear view. Psychodidae SEM view from left. High-resolution SEM image of Psychodidae, drainer moth flies, front view. High-resolution SEM image of Psychodidae, drainer moth flies, whiskers segments. High-resolution SEM image of Psychodidae, drainer moth flies, leg. Health effects. The drain flies, which are commonly found in bathrooms, Clogmia albipunctata, are not known to carry any human diseases, but have been known to be an opportunistic agent of myiasis. However, the subfamily of Phlebotominae does feed on blood with the ability to transmit tropical diseases, and Sycorax silicea can transmit microfilaria. 11. Inhalation of insect fragments may cause asthma. Taxonomy. Notophoricildea zelandiae, previously in Nemopalpus. Mandalea bumersorum. This family has 7 subfamilies that contain more than 2,600 described species. Flea. Flea, the common name for the order Siphonaptera, includes 2,500 species of small flightless insects that live as external parasites of mammals and birds. Fleas live by ingesting the blood of their hosts. Adult fleas grow to about 3 mm, 1/8 inch long, are usually brown, and have bodies that are flattened, sideways or narrow, enabling them to move through their host's fur or feathers. They lack wings. Their hind legs are extremely well adapted for jumping. Their claws keep them from being dislodged, and their mouthparts are adapted for piercing skin and sucking blood. Some species can leap 50 times their body length, a feat second only to jumps made by another group of insects, the superfamily of froghoppers. Flea larvae are worm-like, with no limbs. They have chewing mouthparts and feed on organic debris left on their host's skin. Genetic evidence indicates that fleas are a specialized lineage of parasitic scorpionflies, Mecoptera, sensulato, most closely related to the family Nanochoristidae. The earliest known fleas lived in the Middle Jurassic. Modern-looking forms appeared in the Cenozoic. Fleas probably originated on mammals first and expanded their reach to birds. Each species of flea specializes, more or less, on one species of host. Many species of flea never breed on any other host, some are less selective. Some families of fleas are exclusive to a single host group. For example, the Mecoptera are found only on armadillos, the Ischnopsilidae only on bats, and the Chimaerocylidae only on elephant shrews. The oriental rat flea, Xenopsila cheapus, is a vector of Yersinia pestis, the bacterium that causes bubonic plague. The disease was spread to humans by rodents, such as the black rat, which were bitten by infected fleas. Major outbreaks included the plague of Justinian, about 540, and the Black Death, about 1350, each of which killed a sizable fraction of the world's people. Fleas appear in human culture in such diverse forms as flea circuses, poems, such as John Donne's Erotic, The Flea, works of music, such as those by Modest Mussorgsky, and a film by Charlie Chaplin. Morphology and behavior. Fleas are wingless insects, 1.5 to 3.3 mm, 1/16 to 1/8 inch long, that are agile, usually dark-colored, for example, the reddish-brown of the cat flea with a proboscis, or stylet, adapted to feeding by piercing the skin and sucking their host's blood through their epipharynx. Flea legs end in strong claws that are adapted to grasp a host. 1. Unlike other insects, fleas do not possess compound eyes but instead only have simple eye spots with a single Bickenbeck's lens. Some species lack eyes altogether. 2. Their bodies are laterally compressed, permitting easy movement through the hairs or feathers on the host's body. The flea body is covered with hard plates called sclerites. 1. These sclerites are covered with many hairs and short spines directed backward, which also assist its movements on the host. The tough body is able to withstand great pressure, likely an adaptation to survive attempts to eliminate them by scratching. 3. Fleas lay tiny, white, oval eggs. The larvae are small and pale, have bristles covering their worm-like bodies, lack eyes, and have mouth parts adapted to chewing. The larvae feed on organic matter, especially the feces of mature fleas, which contain dried blood. Adults feed only on fresh blood. 4. Jumping. Their legs are long, the hind pair well adapted for jumping. A flea can jump vertically up to 18 centimeters, 7 inches, and horizontally up to 33 centimeters, 13 and 5, making the flea one of the best jumpers of all known animals, relative to body size, second only to the froghopper. A flea can jump 60 times its length in height and 110 times its length in distance, equivalent to a 1.8 meters, 6 feet adult human jumping 110 meters, 361 feet vertically and 200 meters, 656 feet horizontally. Rarely do fleas jump from dog to dog. Most flea infestations come from newly developed fleas from the pet's environment. 6. The flea jump is so rapid and forceful that it exceeds the capabilities of muscle, and instead of relying on direct muscle power, fleas store muscle energy in a pad of the elastic protein named resilin before releasing it rapidly, like a human using a bow and arrow. 7. Immediately before the jump, muscles contract fleas. Temporal range. Temporal. 6. Their legs are long, the hind pair well adapted for jumping. A flea can jump vertically up to 18 centimeters, 7 inches, and horizontally up to 33 centimeters, 13 and 5, making the flea one of the best jumpers of all known animals, relative to body size, second only to the froghopper. A flea can jump 60 times its length in height and 110 times its length in distance, equivalent to a 1.8 meters, 6 feet adult human jumping 110 meters, 361 feet vertically and 200 meters, 656 feet horizontally. Rarely do fleas jump from dog to dog. Most flea infestations come from newly developed fleas from the pet's environment. 6. The flea jump is so rapid and forceful that it exceeds the capabilities of muscle, and instead of relying on direct muscle power, fleas store muscle energy in a pad of the elastic protein named resilin before releasing it rapidly, like a human using a bow and arrow. 7. Immediately before the jump, muscles contract fleas. Temporal range. Scanning electron micrograph. Scientific classification domain. Eukaryota kingdom. Animalia phylum. Arthropoda class. Insecta clade. Aparaglossata superorder. Panorpida unranked Antliophora order. Siphonaptera. Latreille, 1825. Suborders. Pseudopulicity Sauropteridae tarwinidae. And deform the resilin pad, slowly storing energy which can then be released extremely rapidly to power leg. Extension for propulsion. 8. To prevent premature release of energy or motions of the leg, the flea employs a catch mechanism. 8. Early in the jump, the tendon of the primary jumping muscle passes slightly behind the coxotrochanter joint, generating a torque which holds the joint closed with the leg close to the body. 8. To trigger jumping. Another muscle pulls the tendon forward until it passes the joint axis, generating the opposite torque to extend the leg and power the jump by release of stored energy. 8. The actual takeoff has been shown by high-speed video to be from the tibia and tarsi rather than from the trochanter knees. 7. Dog flea from top larva, egg, pupa and adult. Fleas are holometabolous insects, going through the 4 life cycle stages of egg, larva, pupa, and imago adult. In most species, neither female nor male fleas are fully mature when they first emerge but must feed on blood before they become capable of reproduction. 3. The first blood meal triggers the maturation of the ovaries in females and the dissolution of the testicular plug in males, and copulation soon follows. 9. Some species breed all year round while others synchronize their activities with their host's life cycles or with local environmental factors and climatic conditions. 10. Flea populations consist of roughly 50% eggs, 35% larvae, 10% pupae, and 5% adults. 5. Ceratophyllomorpha, Hystrachopsilomorpha, Pulicomorpha. Pygosylomorpha. Synonyms. Siphonaptera. Life cycle and development. Egg. The number of eggs laid depends on species, with batch sizes ranging from 2 to several dozen. The total number of eggs produced in a female's lifetime, fecundity, varies from around 100 to several thousand. In some species, the flea lives in the host's nest or burrow and the eggs are deposited on the substrate. 9. But in others, the eggs are laid on the host itself and can easily fall off onto the ground. Because of this, areas where the host rests and sleeps become one of the primary habitats of eggs and developing larvae. The eggs take around 2 days to 2 weeks to hatch. 5. Larva. Flea larva. Flea larvae emerge from the eggs to feed on any available organic material such as dead insects, feces, conspecific eggs, and vegetable matter. In laboratory studies, some dietary diversity seems necessary for proper larval development. Blood-only diets allow only 12% of larvae to mature, whereas blood and yeast or dog chow diets allow almost all larvae to mature. 11. Another study also showed that 90% of larvae matured into adults when the diet included non-viable eggs. 12. They are blind and avoid sunlight, keeping to dark, humid places such as sand or soil, cracks and crevices, under carpets and in bedding. 13. The entire larval stage lasts between 4 and 18 days. 14. Pupa. Given an adequate supply of food, larvae pupate and weave silken cocoons after 3 larval stages. Within the cocoon, the larva molts for a final time and undergoes metamorphosis into the adult form. This can take just 4 days, but may take much longer under adverse conditions, and there follows a variable length stage during which the pre-emergent adult awaits a suitable opportunity to emerge. Trigger factors for emergence include vibrations, including sound, heat, in warm-blooded hosts, and increased levels of carbon dioxide, all of which may indicate the presence of a suitable host. 5. Large numbers of pre-emergent fleas may be present in otherwise flea-free environments, and the introduction of a suitable host may trigger a mass emergence. 13. Adult. Once the flea reaches adulthood, its primary goal is to find blood and then to reproduce. 15. Female fleas can lay 5,000 or more eggs over their life, permitting rapid increase in numbers. 16. Generally speaking, an adult flea only lives for 2 or 3 months. Without a host to provide a blood meal, a flea's life can be as short as a few days. Under ideal conditions of temperature, food supply, and humidity, adult fleas can live for up to a year and a half. 16. Completely developed adult fleas can live for several months without eating, so long as they do not emerge from their puparia. Optimum temperatures for the flea's life cycle are 21 to 30 degrees Celsius, 70 to 86 degrees Fahrenheit, and optimum humidity is 70%. 17. Adult female rabbit fleas, Spilopsilus cuniculi, can detect the changing levels of cortisol and corticosterone hormones in the rabbit's blood that indicate it is getting close to giving birth. This triggers sexual maturity in the fleas and they start producing eggs. As soon as the baby rabbits are born, the fleas make their way down to them and once on board they start feeding, mating, and laying eggs. After 12 days, the adult fleas make their way back to the mother. They complete this mini migration every time she gives birth. 17. 7. After 12 days, as soon as the baby rabbit. Optimum temperatures for the flea's life cycle are 21 to 30 degrees Celsius, 70 to 86 degrees Fahrenheit, and optimum humidity is 70%. 17. Adult female rabbit fleas, Spilopsilus cuniculi, can detect the changing levels of cortisol and corticosterone hormones in the rabbit's blood that indicate it is getting close to giving birth. This triggers sexual maturity in the fleas and they start producing eggs. As soon as the baby rabbits are born, the fleas make their way down to them and once on board they start feeding, mating, and laying eggs. After 12 days, the adult fleas make their way back to the mother. They complete this mini migration every time she gives birth. 17. Taxonomy and phylogeny. History. Between 1735 and 1758, the Swedish naturalist Carl Linnaeus first classified insects, doing so on the basis of their wing structure. One of the 7 orders into which he divided them was Aptera, meaning wingless, a group in which as well as fleas, he included spiders, woodlice and myriapods. It wasn't until 1810 that the French zoologist Pierre-André Latreille reclassified the insects on the basis of their mouthparts as well as their wings, splitting Aptera into Thysanura, silverfish, Anaplura, sucking lice, and Siphonaptera, fleas, at the same time separating off the arachnids and crustaceans into their own subphyla. 18. The group's name, Siphonaptera, is zoological Latin from the Greek siphon, a tube, and aptera, wingless. 19. External phylogeny. It was historically unclear whether the Siphonaptera are sister to the Mecoptera, scorpionflies and allies, or are inside that clade, making Mecoptera paraphyletic. The earlier suggestion that the Siphonaptera are sister to the Boridae, snow scorpionflies 2021-22, is not supported. A 2020 genetic study recovered Siphonaptera within Mecoptera, with strong support, as the sister group to Neuroptera, a small, relictual group of Mecopterans native to the southern hemisphere. Fleas and Neuroptera share several similarities with each other that are not shared with other Mecopterans, including similar mouthparts as well as a similar sperm pump organization. 23. Relationships of Siphonaptera per Tyhelka et al. 2020. 23. Anthliophora. Diptera, true flies. Boridae, snow scorpionflies, 30 spp. Neuroptera, southern scorpionflies, 8 spp. Siphonaptera fleas, 2,500 spp. Pistillifera, scorpionflies, hangingflies, 400 spp. Fossil history. Cenozoic flea in amber, circa 20 Maya, is morphologically modern. Pseudophylex wang-yi, a primitive flea from the early Cretaceous of China. Fleas likely descended from scorpionflies, insects that are predators or scavengers. 23. Fossils of large, wingless stem group fleas with siphonate, sucking, mouthparts from the Middle Jurassic 24 to early Cretaceous have been found in northeastern China and Russia, belonging to the families Saurophtheridae and Pseudophyllicidae, as well as Tarwinia from the early Cretaceous of Australia. Most flea families formed after the end of the Cretaceous, in the Paleogene and onwards. Modern fleas probably arose in the southern continental area of Gondwana, and migrated rapidly northwards from there. They most likely evolved with mammal hosts, only later moving to birds. 25. Siphonaptera is a relatively small order of insects. Members of the order undergo complete metamorphosis and are secondarily wingless. Their ancestors had wings which modern forms have lost. In 2005, Medvedev listed 2005 species in 242 genera, and despite subsequent descriptions of new species, bringing the total up to around 2,500 species, 20, this is the most complete database available. The order is divided into 4 infraorders and 18 families. Some families are exclusive to a single host group. These include the Mecoptera, armadillos, Ichneumon bats, and Chimaerocylidae, elephant shrews. 26. Many of the known species are little studied. Some 600 species, a quarter of the total, are known from single records. Over 94% of species are associated with mammalian hosts, and only about 3% of species can be considered to be specific parasites of birds. The fleas on birds are thought to have originated from mammalian fleas. At least 16 separate groups of fleas switched to avian hosts during the evolutionary history of the Siphonaptera. Occurrences of fleas on reptiles is accidental, and fleas have been known to feed on the hemolymph, blood-like body fluid, of ticks. 26. Internal phylogeny. Flea phylogeny was long neglected, the discovery of homologies with the parts of other insects being made difficult by their extreme specialization. Whiting and colleagues prepared a detailed Historychopsilidae Tirabashi, 19. Relationship with host. Flea bites in humans. Fleas feed on a wide variety of warm-blooded vertebrates including dogs, cats, rabbits, squirrels, ferrets, rats, mice, birds, and sometimes humans. Fleas normally specialize in one host species or group of species, but can often feed but not reproduce on other species. Ceratophilus gallinae affects poultry as well as wild birds. 29. As well as the degree of relatedness of a potential host to the flea's original host, it has been shown that avian fleas that exploit a range of hosts only parasitize species with low immune responses. In general, host specificity decreases as the size of the host species decreases. Another factor is the opportunities available to the flea to change host species. This is smaller in colonially nesting birds, where the flea may never encounter another species, than it is in solitary nesting birds. A large, long-lived host provides a stable environment that favors host-specific parasites. 30. Although there are species named dog fleas, C. Tennocephalides canis curtis, 1826, and cat fleas, C. Tennocephalides felis, fleas are not always strictly species-specific. A study in Virginia examined 244 fleas from 29 dogs, all were cat fleas. Dog fleas had not been found in Virginia in more than 70 years, and may not even occur in the U.S., so a flea found on a dog is likely a cat flea, C. Tennocephalides felis. 31, 32. One theory of human hairlessness is that the loss of hair helped humans to reduce their burden of fleas and other ectoparasites. 33. Direct effects of bites. Human foot infested with jigger fleas, Tunga penetrans. In many species, fleas are principally a nuisance to their hosts, causing an itching sensation which in turn causes the host to try to remove the pest by biting, pecking or scratching. Fleas are not simply a source of annoyance, however. Flea bites cause a slightly raised, swollen, irritating nodule to form on the epidermis at the site of each bite, with a single puncture point at the center, like a mosquito bite. 34, 126. This can lead to an eczematous itchy skin disease called flea allergy dermatitis, which is common in many host species, including dogs and cats. 29. The bites often appear in clusters or lines of 2 bites, and can remain itchy and inflamed for up to several weeks afterwards. Fleas can lead to secondary hair loss as a result of frequent scratching and biting by the animal. They can also cause anemia in extreme cases. 126. As a vector. Fleas are vectors for viral, bacterial and rickettsial diseases of humans and other animals, as well as of protozoan and helminth parasites. 35. Bacterial diseases carried by fleas include murine or endemic typhus, 124 and bubonic plague. 36. Fleas can transmit Rickettsia typhi, Rickettsia felis, Bartonella henselae, and the myxomatosis virus. 35. 73. They can carry Hymenoleupiasis tapeworms, 37, and trypanosome protozoans. 35. 74. The Shigo flea or jigger, Tunga penetrans, causes the disease tungiosis, a major public health problem around the world. 38. Fleas that specialize as parasites on specific mammals may use other mammals as hosts. Thus, humans may be bitten by cat and dog fleas. 39. Relationship with humans. In literature and art. Fleas have appeared in poetry, literature, music and art. These include Robert Hooke's drawing of a flea under the microscope in his pioneering book Micrographia published in 1665, 40 poems by Donne and Jonathan Swift, works of music by Giorgio Federico Ghedini and Modest Mussorgsky, a play by Georges Feydeau, a film by Charlie Chaplin, and paintings by artists such as Giuseppe Crespi, Giovanni Battista Piazzetta, and Georges de Latour. 41. John Donne's erotic metaphysical poem, The Flea, published in 1633 after his death, uses the conceit of a flea, which has sucked blood from the male speaker and his female lover, as an extended metaphor for their sexual relationship. The speaker tries to convince a lady to sleep with him, arguing that if the mingling of their blood in the flea is innocent, then sex would be also. 42. The comic poem Siphonaptera was written in 1915 by the mathematician Augustus de Morgan. It describes an infinite chain of parasitism made of ever larger and ever smaller fleas. 43. Robert Hooke's drawing of a flea in Micrographia, 1665. Development of the flea from egg to adult. Antony van Leeuwenhoek, circa 1680. Flea circuses. Flea circuses provided entertainment to 19th century audiences. These circuses, extremely popular in Europe from 1830 onwards, featured fleas dressed as humans or towing miniature carts, chariots, rollers or cannon. These devices were originally made by watchmakers or jewelers to show off their skill at miniaturization. A ringmaster called a professor accompanied their performance with a rapid circus patter. 45. A flea circus, the Go As You Please race, as seen through a magnifying glass, engraved by J. G. Francis, from an article by C. F. Holder in St. Nicholas Magazine, 1886. Carriers of plague. The Great Plague of London, in 1665, killed up to 100,000 people. Oriental rat fleas, Xenopsila cepus, can carry the Coccobacillus yersinia pestis. The infected fleas feed on rodent vectors of this bacterium, such as the black rat, Rattus rattus, and then infect human populations with the plague, as has happened repeatedly from ancient times, as in the plague of Justinian in 541 to 542. 46 outbreaks killed up to 200 million people across Europe between 1346 and 1671. 47 The Black Death pandemic between 1346 and 1353 likely killed over a third of the population of Europe. 48. Because fleas carry plague, they have seen service as a biological weapon. During World War II, the Japanese army dropped fleas infested with Y. pestis in China. The bubonic and septicemic plagues are the most probable form of the plague that would spread as a result of a bioterrorism attack that used fleas as a vector. 49. The Rothschild collection. The banker Charles Rothschild devoted much of his time to entomology, creating a large collection of fleas now in the Rothschild collection at the Natural History Museum, London. He discovered and named the plague vector flea, Xenopsila cepus, also known as the Oriental rat flea, in 1903. 50. Using what was probably the world's most complete collection of fleas of about 260,000 specimens, representing some 73% of the 2,587 species and subspecies so far described, he described around 500 species and subspecies of Siphonaptera. He was followed in this interest by his daughter Miriam Rothschild, who helped to catalog his enormous collection of the insects in 7 volumes. 51. 52. Arthropods portal. Flea treatments. Fleas have a significant economic impact. In America alone, approximately $2.8 billion is spent annually on flea-related veterinary bills and another $1.6 billion annually for flea treatment with pet groomers. $4 billion is spent annually for prescription flea treatment and $348 million for flea pest control. The human flea Pulex irritans, once also called the house flea, 1, is a cosmopolitan flea species that has, in spite of the common name, a wide host spectrum. It is one of 6 species in the genus Pulex. The other 5 are all confined to the Nearctic and Neotropical realms. 2. The species is thought to have originated in South America, where its original host may have been the guinea pig or peccary. 3. Pulex irritans is a holometabolous insect with a 4-part life cycle consisting of eggs, larvae, pupae, and adults. Eggs are shed by the female in the environment and hatch into larvae in about 3 to 4 days. Larvae feed on organic debris in the environment. Larvae eventually form pupae, which are in cocoons that are often covered with debris from the environment, sand, pebbles, etc. The larval and pupal stages are completed in about 3 to 4 weeks when the adults hatch from pupae, then must seek out a warm-blooded host for blood meals. The flea eggs are about 0.5 millimeters in length. They are oval-shaped and pearly white in color. Eggs are often laid on the body of the host, but they often fall off in many different places. The larvae are about 0.6 millimeters in length. They are creamy white or yellow in color. Larvae have 13 segments with bristles on each segment. The larvae feed on a variety of organic debris. The pupa are around 4 by 2 millimeters. After undergoing 3 separate molts, the larvae pupate, then emerge as adults. If conditions are unfavorable, a cocooned flea can remain dormant for up to a year in the pupal phase. The adults are roughly 1.5 to 4 millimeters in length and are laterally flattened. They are dark brown in color, are wingless, and have piercing sucking mouthparts that aid in feeding on the host's blood. Both human flea. Female left, male right. Scientific classification domain: Eukaryota kingdom, Animalia phylum, Arthropoda class, Insecta order, Siphonaptera family, Pulicidae genus, Pullix species, P. irritans binomial name. Pullix irritans. Linnaeus, 1758. Morphology and behavior. Genal and pronotal combs are absent and the adult flea has a rounded head. Most fleas are distributed in the egg, larval, or pupal stages. Relationship with host. Direct effects of bites. Fleas are a pest species to their hosts, causing an itching sensation that results in discomfort and leads to scratching in the vicinity of the bite. Flea bites generally cause the skin to raise, swell, and itch. The bite site has a single puncture point in the center. Bites often appear in clusters or small rows and can remain inflamed for up to several weeks. This species bites many species of mammals and birds, including domesticated ones. It has been found on dogs and wild canids, monkeys in captivity, opossums, domestic cats, wild felids in captivity, chickens, black rats and Norwegian rats, wild rodents, pigs, free-tailed bats, and other species. It can also be an intermediate host for the flea tapeworm Cestodipylidium caninum. Fleas can spread rapidly and move between areas to include eyebrows, eyelashes, and pubic regions. Hair loss as a result of itching is common, especially in wild and domestic animals. Anemia is also possible in extreme cases of high-volume infestations. Vector capabilities. Plague, a disease that affects humans and other mammals, is caused by the bacterium Yersinia pestis. The human flea can be a carrier of the plague bacterium, although it is an exceptionally very poor vector of transmission. Plague is infamous for killing millions of people in Eurasia during the Middle Ages. Without prompt treatment, the disease can cause serious illness or death. Today, human plague infections continue to occur in the western United States, but significantly more cases occur in parts of Africa and Asia. Plague is a very serious illness, but is treatable with commonly available antibiotics. The earlier a patient seeks medical care and receives treatment that is appropriate for plague, the better their chances are of a full recovery. People in close contact with very sick pneumonic plague patients may be evaluated and possibly placed under observation. Preventive antibiotic therapy may also be given, depending on the type and timing of personal contact. Treatment and prevention. Common treatments include body shaving and medicated shampoos and combing. When preventing fleas, it is important to treat both the host and the environment. Household pets should be treated to prevent being infested. Carpets, and other floor surfaces, should be shampooed and vacuumed regularly to prevent fleas. When dealing with an infestation, bedding and clothing that may have been exposed should be washed thoroughly in hot water and kept away from other materials that have not been exposed. In extreme cases, extermination by a professional is necessary. Phylogeny and distribution. Pullix irritans originated in Central and South America, where it evolved with only one host. 5. Despite the origins of P. irritans, it is now a cosmopolitan species, most likely due to the fur trade after the 18th century, and pet transport in more recent decades. 5. 6. Due to the cosmopolitan distribution of this group, research has been conducted to show that P. irritans individuals, when sampled from Spain and Argentina, only showed significant differences when comparing molecular data. 7. The use of morphometric data, which is often the method for flea identification, showed no significant levels of divergence between the two populations. Combining both morphological and molecular data has identified the possibility of cryptic species. This indicates a need to reconsider taxonomic classifications of these fleas. Studies in Europe and the Mediterranean also found this to be true. Human and cat fleas from Europe and the Mediterranean were studied and show such great variation within their own species that their taxonomy should be reevaluated based on mitochondrial gene heterogeneity. 6. References. 1. The premise. The insect order Neuroptera, or net-winged insects, includes the lacewings, mantidflies, antlions, and their relatives. Neuroptera. The insect order Neuroptera, or net-winged insects, includes the lacewings, mantidflies, antlions, and their relatives. The order consists of some 6,000 species. 1. Neuroptera is grouped together with the Mecoptera alderflies, fishflies, and dobsonflies, and Rhaphidioptera snakeflies, in the unranked taxon Neuropterida, once known as Planopenia. Adult Neuropterans have 4 membranous wings, all about the same size, with many veins. They have chewing mouthparts, and undergo complete metamorphosis. Neuropterans first appeared during the Permian period, and continued to diversify through the Mesozoic era. 2. During this time, several unusually large forms evolved, especially in the extinct family Calligrammatidae, often called the butterflies of the Jurassic, for their large, patterned wings. 3. Neuropterans are soft-bodied insects with relatively few specialized features. They have large lateral compound eyes, and may or may not also have ocelli. Their mouthparts have strong mandibles suitable for chewing, and lack the various adaptations found in Most other holometabolan insect groups. They have 4 wings, usually similar in size and shape, and a generalized pattern of veins. 4. Some neuropterans have specialized sense organs in their wings, or have bristles or other structures to link their wings together during flight. 5. The larvae are specialized predators, with elongated mandibles adapted for piercing and sucking. The larval body form varies between different families. Neuroptera. Temporal range, Permian to recent. Green lacewing. Scientific classification domain, Eukaryota kingdom, Animalia phylum, Arthropoda class, Insecticlade, Neuropterida order, Neuroptera. Linnaeus, 1758. Clades. See taxonomy. Anatomy and biology. Larva of Osmilus fulvicephalus, Osmilidae. Larva of Cicera sp. Ciceridae. Depending on the nature of their prey. In general, however, they have 3 pairs of thoracic legs, each ending in 2 claws. The abdomen often has adhesive discs on the last 2 segments. 5. The larvae of most families are predators. Many chrysophids, hemerobiids and coniopterygids eat aphids and other pest insects, and some have been used for biological control either from commercial distributors, but also abundant and widespread in nature. 6. 7. Larvae in various families cover themselves in debris, including other insects, living and dead. 8. As camouflage, taken to an extreme in the ant lions, which bury themselves completely out of sight and ambush prey from pits in the soil. Larvae of some Ithonidae are root feeders, and larvae of Ciceridae are aquatic, and feed on freshwater sponges. A few mantispids are parasites of spider egg sacs. As in other holometabolic orders, the pupal stage is enclosed in some form of cocoon composed of silk and soil or other debris. The pupa eventually cuts its way out of the cocoon with its mandibles, and may even move about for a short while before undergoing the molt to the adult form. 5. Adults of many groups are also predatory, but some do not feed, or consume only nectar. Beetles, wasps, and some lake flies parasitize neuropteran larvae. Life cycle of lacewings. Life cycle and ecology. Evolution. One of the butterflies of the Jurassic, Sophogramma Lee calligrammatidae. Neuropterans first appeared near the end of the Permian period, as shown by fossils of the Permithonidae from the Tunguska Basin in Siberia and a similar fauna from Australia. 2. Patterned wing of Paleogene 49 Maya, fossil Palaeocycops maringerae, Ithonidae. The osmilids are of Jurassic or early Cretaceous origin and may be the most ancient of the neuropteran groups. 9th. The extinct osmilid Protosmilus is fossilized in Middle Eocene Baltic amber. 10th. The genus Burmalion is described from 2 fossils of Cenomanian age Burmese amber, implying crown group radiation in the early Cretaceous or earlier. 11. 12. The family Calligrammatidae lived from the Jurassic to Aptian, lower Cretaceous periods. 13. Ithonidae are from the Jurassic to recent, and the extinct lineages of the family were widespread geographically. 14. Following the end of the Cretaceous period, the diversity of neuropterans appears to have declined. 15. Phylogeny. Molecular analysis in 2018 using mitochondrial rRNA and mitogenomic data places the Mecoptera as sister to Neuroptera, and Rhaphidioptera as sister to this combined lineage, though these results were considered tentative. 16. 9. The fossil record has contributed to the understanding of the group's phylogeny. 1. 17, 18, 19. Relationships within the Myrmeleontiformia are still in flux. 20. A phylogenomic analysis published in 2023 confirmed the topology of the neuropterid orders and found the relationships between the families of Neuropterida as shown in the following phylogenetic tree. 21. The fossil genus Mesohemerobius Ping, 1928 from the late Jurassic early Cretaceous of China has been treated as incertae sedis within Neuroptera, while the fossil families Permobrothidae and Permithonidae are treated as a sister group to clade Edoneuroptera formed by Neuroptera plus Megaloptera. 22. In human culture. The use of Neuroptera in biological control of insect pests has been investigated, showing that it is difficult to establish and maintain populations in fields of crops. 24. 5 species of Neuroptera are among 1681 insect species eaten by humans worldwide. 25. The New Guinea Highland people claim to be able to maintain a muscular build and great stamina despite their low protein intake as a result of eating insects including Neuroptera. Neuroptera. The insect order Neuroptera, or net-winged insects, includes the lacewings, mantid flies, antlions, and their relatives. The order consists of some 6,000 species. 1. Neuroptera is grouped together with the Megaloptera alderflies, fishflies, and dobsonflies, and Rhaphidioptera snakeflies, in the unranked taxon Neuropterida, once known as Planopenia. Adult neuropterans have 4 membranous wings, all about the same size, with many veins. They have chewing mouthparts, and undergo complete metamorphosis. Neuropterans first appeared during the Permian period, and continued to diversify through the Mesozoic era. 2. During this time, several unusually large forms evolved, especially in the extinct family Calligrammatidae, often called the butterflies of the Jurassic, for their large, patterned wings. 3. Neuropterans are soft-bodied insects with relatively few specialized features. They have large lateral compound eyes, and may or may not also have ocelli. Their mouthparts have strong mandibles suitable for chewing, and lack the various adaptations found in most other holometabolan insect groups. They have 4 wings, usually similar in size and shape, and a generalized pattern of veins. 4. Some neuropterans have specialized sense organs in their wings, or have bristles or other structures to link their wings together during flight. The larvae of most families are predators. Many chrysophids, hemerobiids and coniopterygids eat aphids and other pest insects, and some have been used for biological control either from commercial distributors, but also abundant and widespread in nature. 6. 7. Larvae in various families cover themselves in debris, including other insects, living and dead. 8. As camouflage, taken to an extreme in the ant lions, which bury themselves completely out of sight and ambush prey from pits in the soil. Extreme in the ant lions. Larvae of some Ithonidae are root feeders, and larvae of Tsetsefly are aquatic, and feed on freshwater sponges. A few mantispids are parasites of spider egg sacs. As in other holometabolic orders, the pupal stage is enclosed in some form of cocoon composed of silk and soil or other debris. The pupa eventually cuts its way out of the cocoon with its mandibles, and may even move about for a short while before undergoing the molt to the adult form. Mecoptera. Mecoptera, from the Greek, mekos equals long, ptera equals wings, is an order of insects in the superorder Holometabola with about 600 species in 9 families worldwide. Mecopterans are sometimes called scorpionflies after their largest family, Panorpidae, in which the males have enlarged genitals raised over the body that look similar to the stingers of scorpions, and long beak-like rostra. The Bitacidae, or hangingflies, are another prominent family and are known for their elaborate mating rituals, in which females choose mates based on the quality of gift prey offered to them by the males. A smaller group is the snow scorpionflies, family Boridae, adults of which are sometimes seen walking on snowfields. In contrast, the majority of species in the order inhabit moist environments in tropical locations. The Mecoptera are closely related to the Siphonaptera fleas, and a little more distantly to the Diptera true flies. They are somewhat fly-like in appearance, being small to medium-sized insects with long slender bodies and narrow membranous wings. Most breed in moist environments such as leaf litter or moss, and the eggs may not hatch until the wet season arrives. The larvae are caterpillar-like and mostly feed on vegetable matter, and the non-feeding pupae may pass through a diapause until weather conditions are favorable. Most breed The Mecoptera are closely related to the Siphonaptera fleas, and a little more distantly to the Diptera true flies. They are somewhat fly-like in appearance, being small to medium-sized insects with long slender bodies and narrow membranous wings. Most breed in moist environments such as leaf litter or moss, and the eggs may not hatch until the wet season arrives. The larvae are caterpillar-like and mostly feed on vegetable matter, and the non-feeding pupae may pass through a diapause until weather conditions are favorable. Early Mecoptera may have played an important role in pollinating extinct species of gymnosperms before the evolution of other insect pollinators such as bees. Adults of modern species are overwhelmingly predators or consumers of dead organisms. In a few areas, some species are the first insects to arrive at a cadaver, making them useful in forensic entomology. 9. Diversity. Mecopterans vary in length from 2 to 35 mm, 0.1 to 1.4 in. There are about 600 extant species known, divided into 34 genera in 9 families. The majority of the species are contained in the families Panorpidae and Bitacidae. Besides this, there are about 400 known fossil species in about 87 genera, which are more diverse than the living members of the order. 10. The group is sometimes called the scorpionflies, from the turned-up tail of the male's genitalia in the Panorpidae. 11. Distribution of Mecopterans is worldwide. The greatest diversity at the species level is in the Afrotropic and Palearctic realms, but there is greater diversity at the generic and family level in the Neotropic, Nearctic and Australasian realms. They are absent from Madagascar and many islands and island groups. This may demonstrate that their dispersal ability is low, with Trinidad, Taiwan and Japan, where they are found, having had recent land bridges to the nearest continental land masses. 10. Taxonomic history. The European scorpionfly was named Panorpa communis by Linnaeus in 1758. 12. The Mecoptera were named by Alpheus Hyatt and Jenny Maria Arms in 1891. 13. The name is from the Greek, mekos meaning long, and ptera meaning wings. 14. The families of Mecoptera are well accepted by taxonomists, but their relationships have been debated. In 1987, R. Willman treated the Mecoptera as a clade, containing the Boridae as sister to the Meroptera. 15. But in 2002 Michael F. Whiting declared the Mecoptera as defined as paraphyletic, with the Boridae as sister to another order, the Siphonaptera fleas. 16. Fossil history. Among the earliest members of the Mecoptera are the Nanochoristidae of Upper Permian age. Fossil Mecoptera become abundant and diverse during the Cretaceous, for example in China. 17. Where panorpids such as Drosophilidae, hangingflies Bitacidae and Orthophlebeidae, and Orthophlebeidae have been found. 18. 19. Extinct Mecoptera species may have been important pollinators of early gymnosperm seed plants during the late Middle Jurassic to mid-Mecoptera. Temporal range. Panorpa communis, male. Scientific classification domain, Eukaryota kingdom, Animalia phylum, Arthropoda class, Insecta, unranked, Antlyphora. Order, Mecoptera. Packard, 1886 Comstock, 1895. Families. Apteropanorpidae. Boridae, snow scorpionflies, Choristidae. Choristopsychidae, 1. Englathaumatidea, 2. Eomeropodidae. Meropodidae, earwig flies. Mesopsychoidea equals Anertopsychina, 3. Evolution and phylogeny. Early Cretaceous periods before other pollinating groups such as the bees evolved. These were mainly wind-pollinated plants, but fossil. Mecopterans had siphon feeding apparatus that could have fertilized these early gymnosperms by feeding on their nectar and pollen. The lack of iron enrichment in their fossilized proboscis rules out their use for drinking blood. 11 species have been identified from 3 families: Mesopsychidae, Anertopsychidae, and Pseudopolycentropididae within the clade Anertopsychina. Their lengths range from 3 mm (0.12 in Parapolycentrupus burmaticus to 28 mm (1.1 in Lichnomysocyte glorii. The proboscis could be as long as 10 mm (0.39 in). It has been suggested that these Mecopterans transferred pollen on their mouthparts and head surfaces, as do bee flies and hoverflies today, but no such associated pollen has been found, even when the insects were finely preserved in Eocene Baltic amber. They likely pollinated plants such as Cataniaceae, Chirolepidiaceae, and Natales, which have ovulate organs that are either poorly suited for wind pollination or have structures that could support long proboscis fluid feeding. 2021. The Anertopsychina were the most diverse group of Mecopterans in the latest Permian, taking the place of the Permachoristidae to the Middle Triassic. During the late Triassic through the Middle Jurassic, Anertopsychina species were gradually replaced by species from the Parachoristidae and Orthophlebeidae. Modern Mecopteran families are derived from the Orthophlebeidae. 22. Mesopsychidae. Jurasymbrophlebe. A Ginkgofolia, Symbrophlebeidae, reconstruction, Jurassic, China. Jurassipanorpa. Sticta-panorpidae, Jurassic, China. Forewing of Dinokanaga andersoni. Dinopanorpidae. Eocene, Washington State. External relationships. Mecoptera have special importance in the evolution of the insects. 2 of the most important insect orders, Lepidoptera butterflies and moths, and Diptera true flies, along with Trichoptera caddisflies, probably evolved from ancestors belonging to, or strictly related to, the Mecoptera. Evidence includes anatomical and biochemical similarities as well as transitional fossils, such as Promodonidaris and Choristodonidaris, which lie between the Mecoptera and Diptera. The group was once much more widespread and diverse than it is now, with 4 suborders during the Mesozoic. 23. It is unclear as of 2020 whether the Mecoptera form a single clade, or whether the Siphonaptera fleas are inside that clade, so that the traditional Mecoptera taxon is paraphyletic. However, the earlier suggestion that the Siphonaptera are sister to the Boridae 24, 16, 25 is not supported. Instead, there is the possibility that they are sister to another Mecopteran family, the Nanochoristidae. The 2 possible trees are shown below. 26. A Mecoptera clades in boldface is paraphyletic, containing Siphonaptera. 26, 27. Male Panorpa dubia. A. Body in lateral view. B. D. Male genital bulb and gonostyle. B. Dorsal view. C. Ventral view. D. Lateral view. EP. Apandrium. GCX. Gonococcite. GS. Gonostylus. HV. Hypovalva. HYP. Hypandrium. Scale bars represent 3 mm in A, 1 mm in BD. Biology. Morphology. Mecoptera are small to medium-sized insects with long beak-like rostra, membranous wings and slender, elongated bodies. They have relatively simple mouthparts, with a long labium, long mandibles and fleshy palps, which resemble those of the more primitive true flies. Like many other insects, they possess compound eyes on the sides of their heads, and 3 ocelli on the top. The antennae are filiform, thread-shaped, and contain multiple segments. 23, 29. The fore and hind wings are similar in shape, being long and narrow, with numerous cross veins, and somewhat resembling those of primitive insects such as mayflies. A few genera, however, have reduced wings, or have lost them altogether. The abdomen is cylindrical with 11 segments, the first of which is fused to the metathorax. The cerci consist of 1 or 2 segments. The abdomen typically curves upwards in the male, superficially resembling the tail of a scorpion, the tip containing an enlarged structure called the genital bulb. 23, 29. The caterpillar-like larvae have hard sclerotized heads with mandibles, jaws, short true legs on the thorax, prolegs on the first 8 abdominal segments, and a suction disc or pair of hooks on the terminal 10th segment. The pupae have free appendages rather than being secured within a cocoon, they are exarate. 29. Ecology. A panorpid scorpionfly feeding on a dead insect. Mecopterans mostly inhabit moist environments, although a few species are found in semi-desert habitats. Scorpionflies, family Panorpidae, generally live in broadleaf woodlands with plentiful damp leaf litter. Snow scorpionflies, family Boridae, appear in winter and are to be seen on snowfields and on moss, the larvae being able to jump like fleas. Hanging flies, family Bitacidae, occur in forests, grassland and caves with high moisture levels. They mostly breed among mosses, in leaf litter and other moist places, but their reproductive habits have been little studied, and at least one species, Nanochorostophilpati, has aquatic larvae. 10. Adult Mecopterans are mostly scavengers, feeding on decaying vegetation and the soft bodies of dead invertebrates. Phanorpa raids spider webs to feed on trapped insects and even the spiders themselves, and hanging flies capture flies and moths with their specially modified legs. Some groups consume pollen.